So I will be recording this and posting the recording as well. And a quick reminder before we get started that we will be going over that one card definition term sheet. And so I'm going to be cold calling you for answers on that one. All right. So today we want to focus on epidemiology. It's really a shame that we're not in person because I think this is like one of the more fun chapters in the class, but it really has a lot of relevance to what we talked about last class, which is to say we're focused on diseases. Now, of course, epidemiology has a different kind of scope and specific focus than pathogenesis. And unlike pathogenesis, which I call the chapter of many definitions, this is the chapter of fun math. I really do think the math is fun, and I think that you may agree with me. But if we compare these two different ways of approaching disease in people, pathogenesis is really focusing on the molecular details of how pathogens interact with their hosts. And epidemiology is focused on how disease goes through populations. So it's looking at patterns and much larger data sets. Just to make sure that we understand how these different fields approach concepts in disease, I'm going to ask you some questions, and you're going to answer the questions by putting your answers into the chat. Okay. So if you wanted to describe the virulence factors of this pathogen, Borrelia burgdorferi, which is the causative agent of Lyme disease, would it make more sense to study it as from a pathogenesis standpoint or from an epidemiological one? Put your answer into the chat. So you need to find the chat. Hello, Eric. Welcome. I can read you. Right now we are putting answers into the chat. The first question I've asked you is, if you were studying the virulence factors of this organism, Borrelia burgdorferi, would it make more sense to study it as a pathogenicist or as an epidemiologist? So everyone needs to put their answers into the chat. And we'll wait until we get them all in. Awesome. Okay. I agree. Pathogenesis for sure. What if we wanted to determine what's the percentage or the fraction of the population on campus that has contracted coronavirus? Is this an epidemiological question, or is this something that we would discuss as under the purview of pathogenesis? I agree. I think it's an epidemiological question for sure. What if we wanted to identify the specific cell receptors HIV uses to enter T cells during infection? Right. Yes, pathogenesis. And last question, what if we were tracking how many people usually die of flu annually in the US? Hey, Eric, I can see your chats. Are you able to hear us? Yes, epidemiological question, indeed. So if we think about what the goal is overall of the epidemiological field, what would you say? Right? Like just a quick sentence into the chat that kind of describes the goal of this field, if you could kind of summarize it.
Can you ask the question again?
Yes. The question is, what is the goal of epidemiology? I mean, that's kind of a big question. I realize that, but if you could sort of summarize it, it doesn't necessarily have to be the best answer on the planet, but what is the goal of epidemiology? What is the, what are people trying to do when they're practicing in this field? Eric, can you unmute? Or can you hear me? Right. Okay. So I'm just going to read some of your answers. I think they're really good. Julia said, study the spread of study of the spread, control, and prevention of disease in populations. And Eunice said, study patterns of disease. Oops. My chat keeps scrolling. Study patterns of disease focusing on the control of the spread. Yeah. Okay. So Eric, I don't know if you can hear me because, and I can read your chats, but if you can hear me, can you say yes and put that in the chat so I know that me speaking is helpful? Okay, great. So just to reiterate what was in the class announcements and in the rave alert that went out earlier, the snow caused the campus to go into lockdown and we are not on campus. I am, for that reason, Zooming the class. So there's nobody on campus. I'm not on campus. And I'm so sorry, Eric, that you are on campus because you are not going to be able to get into the building, unfortunately. You probably can get into the Starbucks across the street if you want to, but I am going to be recording this and posting it if that's easier. So it's kind of up to you. I know, sorry that you didn't get the messages. That sucks. All right. So we're not going to do a breakout because we're a small enough group. We're just going to do this all together. And we're going to do this kind of like we did the last one. These are terms from our term sheet. And we can see that there are some individual terms. There are some compared terms. There's some even like 3 terms at a time. So what I'm going to do is I'm going to ask each of you in turn to, to pick one. This is, okay, there we go. To pick one of these and define it, and hopefully we can all guess. So the guessing will happen in the chat. It's not going to be out loud, but I will call on you individually, and you're going to give me a definition or multiple definitions to try to get us to guess one of these line items. And then people will put their answers into the chat. As always, if I ask you to do one of these and you just kind of draw a blank, you can always say pass. But let's begin with Julius. Don't tell us what you are going to define, but give us you can unmute and give us a definition or a couple of contrasting definitions that will allow us to try to guess one of these.
Oh my gosh, you put me on the spot.
And you're always allowed to pass, but it's okay if you want to just take a stab at it anyway.
It's when you get sick in a hospital setting.
Okay, great. He says when you get sick in a hospital setting, put your answers into the chat. What do you think he's talking about, guys? You define one of these line items. And now read the chat and tell us, Julius, are people right?
That's correct.
Excellent job. All right, now we know what we're doing. Fantastic. Thanks for being my first guinea pig. All right, next up, we're going to have Kate go ahead and pick one of these and define it. If you want to, you can always pass.
Okay, let me see here. Just pull up my sheet. Yeah, so this would be like immunity of most of the population through natural or vaccinations.
Great. What do you think, guys? Put the answers into the chat. What do you think she's talking about? This is the one we just did. All right. We got some answers coming in. All right, Kate, are they right?
Yes.
Excellent. Herd immunity it is. Good work. Okay. Fantastic. You guys are excellent at using those reaction emojis. Fantastic.
They're fun.
They are very fun. All right. Next we have Taylor. Taylor, go ahead and if you want to, give us some definitions so we can guess one of these line items.
Okay. The first one is fraction of population that contracts a disease over time in a defined time period versus fraction of population with a specific disease at a given time in defined time period.
All right, guys, what do you think she is defining for us? Write your answers into the chat and Taylor's going to tell us if we got it. Fraction of population that contracts a disease in a defined time period versus fraction of population that has that disease in a defined time period. So am I correct in saying, Taylor, that the correct answer has not yet been stated?
Yes.
Because I think you did a great job. All right, guys, try again. Those were good guesses, but they're not correct.
Can you say it again?
She said fraction of the population that contracts a disease in a given time period versus fraction of the population that has the disease in a given time period.
Eunice got it.
Yes, good job, Eunice. All right, incidence versus prevalence. How are we feeling about that one? Good. Okay, Eunice, I'm coming for you. Go ahead and unmute. You are always allowed to pass, but if there's one of these you'd like to define for us, go ahead. Are you able to unmute, Eunice? You can also put your clues into the chat if you'd rather not do it. She's going to pass. Okay. Darsh, you're up next. Are you going to pass or would you like to do one?
I'm gonna pass.
No problem. Okay, so that means we're back to Julius. Is there one you'd like to do?
Sure. It's the cases of the specific notifiable disease and the number of death, and the number of death of people affected in relation to the total population, and then the number of death in relation to the population.
Okay, say that one more time.
Sorry, it's okay. Number of death. Oh, I put my notes in very short sentences. So the first one is number of death, and then
Okay.
the second one is the number of death affected in relation to the total population. So number of death over the total number of population.
Okay, so Kate is guessing this one here. To me, it sounds like you defined this versus that. Does that sound right?
And yes, and then the cases of the specific disease.
Okay, so cases of specific disease versus number of deaths from that disease versus deaths as a fraction of the total population. Yes, good job. I know that was a hard one. Good work. Okay, Kate, you're always allowed to pass because we've already come through you, but if there's one of these that you feel like you want to do, you're more than welcome.
Okay, let me see here. So this is like, so this will tell us where you can catch them versus when you are at risk.
Okay, what do you think she's talking about, guys? This will tell us where, okay, so we've got an answer from
Yeah.
Julius. Julius is correct. So where we're likely to be exposed versus how we're likely to be exposed, be another way to say this, but where we're likely to catch it versus what's the risk. Good job. Okay, now we just have a couple left. So what I'd like to do is actually have you write your answers into the chat for this one. Okay, endemic versus epidemic versus pandemic, right into the chat how you would define these terms. Let's have everyone do this. Okay, so Eunice says a disease that's constantly present in a population. That's endemic. Okay, great. Who wants to give an epidemic? Epidemic. So endemic is a disease constantly present in the population. That's from Eunice. Epidemic or pandemic. Who wants to give me one of those? Write your answer into the chat. Okay, epidemic acquired by many hosts in a short time. And or unusually large caseload. Yes, and then finally, pandemic is going to be an epidemic that is global, right? Yes, pandemic is a widespread illness, really very widespread over most of the globe. Okay, and last but not least, we have mechanical vector transmission versus biological vector transmission. Is there anyone who'd like to give the definition of these, to compare and contrast them out loud, verbally? Raise your hand. Go ahead, Julius.
So for the mechanical, in an easier term, I think it's the pathogen on feet. That's something that can walk on you, like a direct transmission. And then for the biological vector, that is something when you get bitten,
Yes.
and they put like a venom or something in you.
Yes. So in this context, vectors really referring usually to insects. And are you catching it because the insect brought it on its body and walked on your food, let's say with its little feet? So it landed on some feces at an outhouse and then got the pathogen on its feet and then landed on your hamburger at a picnic and walked on it. That's mechanical. Or is the insect biting you and injecting the pathogen into your body, subcutaneously or, through your skin, and using is the pathogen also inside of the insect? So it's inside this insect's salivary glands, for example. Fabulous. Okay, so epidemiology really is the study of the spread, control, and prevention of diseases in populations. And we can probably all think of things that we have learned how to do or things that we take for granted as infrastructure that we just benefit from, that were informed by epidemiological principles. Can you think of any? Put them into the chat. I will read them out as you put them in. You want to think about epidemiology looks at whole populations and risk factors and how we can alleviate that disease risk. What kinds of things were you taught to do? Probably because an epidemiologist said, let's teach everybody or to encourage everyone to do this. What kind of infrastructure has been built specifically with the idea of reducing disease transmission in mind? Okay, great. Vaccine campaigns, good example. Hand washing, great example. Being encouraged to wear a mask in certain situations at certain times. Quarantining, yes, fabulous. Good examples, guys. So here's a few of mine. You covered some of them. Some of the infrastructure things, they're sort of more subliminal in a way, right? We just live with them. We don't think about them too much, but treating wastewater or treating drinking water is an important way to reduce disease exposure, and having good waste removal, not having trash and other things sitting around. So reservoirs, as was defined earlier, is the location where the pathogen is normally found. And potential reservoirs include other people, animals, food, the environment. That includes air, water, and soil. The point, like why an epidemiologist cares about a reservoir is why? What's the importance of a reservoir? If you're an epidemiologist, why would you want to know that? Put that into the chat. Okay, the source of the infection potentially is what Julius wrote. Anyone have anything they want to add to that? Being able to control it. Yeah, I would say those are two good ways of answering this question. Stop it at the source. Yeah, I agree. Another way to look at it is, maybe it's not, maybe knowing where something is found is not actually going to allow you to completely eliminate it. For example, botulism and tetanus are caused by spores in the soil. It's really hard to eliminate that because you would have to like do a scorched earth policy, but it does allow us to mitigate the risk. If you know that you can get tetanus endospores inside of a deep cut, then you are more likely to pay attention. If you get a deep cut, get a tetanus shot every 10 years, check your tetanus vaccine status if you get a deep cut, that kind of stuff. You're more likely to prepare food in a way that would destroy endospores because you're worried about botulism. You are more likely to avoid feeding unsterilized food to babies because they can't handle it. That might have botulism spores in it, like raw honey, for example. All right, so it determines how likely you are to be exposed and what kinds of behaviors might help to reduce your risk. The coronavirus reservoir, of course, where are you most likely to encounter? What's the reservoir for coronavirus? Pretty much most of the time. Yeah, that's right. It is other people. Yeah. Now, it originally did come from animals, probably lots of different viruses come from animals in the initial stage, and that's probably going to happen again. But right now, you're much more likely to be exposed because of meeting other people rather than running into the animal. All right, so for each major mode of transmission, give an example. So I'm going to list them and you're going to give me an example. Direct contact transmission. What's an example of direct contact? Touching, okay, kissing, HPV, handshake, yes, good, very good. Okay, so we're basically having like direct contact. It's kind of a, what about an example of fecal oral? What kind of behavior or situation might allow you to contract something that's normally transmitted by this route of transmission? Yes, drinking dirty water. Yeah, that's very true. Any other examples, guys? Contaminated foods prepared by people who didn't wash their hands. Exactly. So getting water in your mouth and swallowing it from some kind of water source that has had people or animals in it and has not been treated, okay? Or having food that was prepared by somebody who didn't wash their hands. Droplet transmission. Examples of, yeah, inhaling some, someone coughing, someone talking, someone singing. Here are some, situations where you might be at higher risk if you're in a crowded arena with tons and tons of people, or if you're sitting down and sitting next to people for long periods of time in really close quarters. What about fomite? Fomite transmission. Can you give me an example of that? Yeah. I used Kleenex tissue. A doorknob, right. So, yeah, it's touching objects like someone's cell phone. Yep, exactly. What about zoonotic transmission? Any thoughts? Yeah, getting animal bites or insect bites. Also touching animals sometimes, like milking cows can give you cowpox. Parenteral. What might be an example of parenteral? Yeah. Needle sharing, needle stick, or some kind of wound, really any kind of wound, right? And then vector transmission would be that one that we talked about. Involves insects, housefly, walking on fecal matter, mosquitoes biting you, that kind of stuff. Yes, for parenteral scrapes and cuts. Okay. So let's have a quick look at vector transmission. We defined it earlier. I just want to revisit it. The vector is, of course, insects, usually fleas, ticks, mosquitoes. And the mechanical vector transmission is when the insect is bringing the pathogen on its body, on the outside of its body, and somehow transmitting it to you. Okay, so which of these pictures on the left or the right do you think depicts mechanical transmission? Mechanical transmission, left or right? Oh, we've got a 50-50 split. Excellent. Okay. Well, I'm not going to give the answer yet since we are not in agreement. Mechanical is when the insect carries the pathogen on its body, such as on its feet. And biological is when the pathogen reproduces inside of the insect and then it is introduced into you. And so I would say for sure the mechanical picture is this one, right? Because it's transmitting the pathogen passively. It's on its body and it's just probably it walked on some manure, wandered around in an outhouse or on some kind of surface that had something on it. And now it's walking on your food and you're going to swallow it because it was passively transmitted on the body of the insect. Whereas when you get malaria or yellow fever or chikungunya or something that's transmitted by a mosquito bite, the pathogen has actually had part of its life cycle inside the mosquito, and it's transmitting it to you through its saliva when it punctures your skin. Does that make sense? Yes? No? Fantastic. Okay. I need to hear from the people who originally had the other answer to see if it makes sense. So let me ask you directly. Okay, good. Excellent. All right. So now we're going to start with some of the fun math. So morbidity we defined it earlier, is basically just the total caseload in a defined population in time. And it's just a number, it's just a raw number like 12 or 1,257. Incidence, on the other hand, is a percentage or a decimal fraction or a ratio. And what it is the fraction of the population that has contracted the disease in a defined time period. In other words, in that time period, how many people are new cases? They were newly diagnosed or newly acquired the disease. As a fraction of the population, usually it's expressed as a percentage. So whatever that population is, the new cases divided by population times 100 would give you the percentage. What's the difference between incidence then, which is the fraction of new cases over the total population, and prevalence? How would you define prevalence different from incidence? Put that into the chat. Okay, good, Taylor. Yes. So Taylor says the fraction of the population that has a specific disease at a given time in a defined time period. So it's very similar, except that it's looking at total cases. So it's not looking at, did you contract this disease in this time period? It's just, do you have it in this time period? And this is likely to be a larger number, because if there's 10 people who have a cold on Tuesday, but only 2 of them caught the cold that day and the rest had it the previous day, then we have, 10 over whatever that population is our prevalence, but 2 over whatever that population is our incidence. So does that distinction make sense to everybody? New cases over population versus total cases over population? Great. Let's see if it really made sense. So let's suppose October 25th, 2021, there was a campus with 30,000 students. There's our population, total population. There were 600 cases of the common cold, but of those, 150 had been contracted in the last 24 hours. So let's start with morbidity. What's our morbidity in this case? What is morbidity? Put it into the chat. Remember that morbidity is just total caseload. It's a raw number. It's not a fraction. It's not the population. Morbidity is just the total caseload. What is the total caseload? How many cases do we have? It's not 30,000. That's our total population. And it's not 150. Those are our new cases. Julius is right. Eunice is right. That's right, Taylor. 600 is our morbidity. Okay. How many people have it? Now we need to calculate incidence and prevalence. So let's start with incidence. Incidence is going to be new cases divided by population times 100 expressed as a percentage. So you can do that math on your phone or on your computer and then put it into the chat. If you are feeling so brave as to show your work, go right ahead. Why are we doing this? We're doing this so that we can get good at just answering it on the fly, because you were kind of on the spot right now. I mean, I didn't give you a lot of prep. That's what it's going to be like on the test. So we're just practicing. So what you want to do for this calculation is you want to take, for incidence, we want new cases divided by total population of all the people, whether they're sick or not, times 100. Yeah. Just working it, just getting there, going through the motions, all good in the hood. So what is our answer? And go ahead and show your work if you want. I'll wait for everybody. Okay, good. Good. All right, nice work, Kate. So Kate has shown us her work, and it's consistent with other people's. 150 divided by 30,000 is equal to 0.005. And if you want to express that as a percentage, you multiply by 100, and that ends up being 0.5, but you'd want to put a percent next to it so that we're clear it's 0.5%. Beautiful. Okay, what about prevalence? How would I calculate that? Prevalence is total cases over population times 100 to get the percentage. What is that going to be? What is our total cases divided by population expressed as a percentage? Yep. Great. All right, so we got our morbidity of 600. That's our total caseload. Incidence, new cases divided by population, 0.005. If we want it as a percentage, we multiply by 100, 0.5%. Prevalence, total caseload divided by 30,000, which is our total population, is equal to 0.02 as a percentage times 100, that's 2%. Good, good. Okay. So earlier we also defined mortality and mortality rate. I'm leaving morbidity up here so that we can compare when we look at these definitions, because sometimes people get these two metrics confused. I'm also going to define case fatality rate, even though it's not in your curriculum, but it's fun to know. Okay, so mortality is total deaths. Like morbidity, it's a raw number. It's not a fraction, it's just a number. How many people have died? Mortality rate, on the other hand, is the fraction of people who died out of the total population. It's not the total sick population, it's just the total population period, okay? So it's the fraction of the population that has died of a particular disease, usually expressed as a percentage. This is different from the case fatality rate, which is the number of deaths over total cases. Again, you will not be tested on this one in purple. Let me give you a sense of the difference between what these numbers, these fractions tell us. Mortality rate gives us a very crude idea of your risk of dying from a disease because of where you live. Like if the population you're looking at is the county or the state of Washington, okay? Then looking at the mortality rate from whatever disease in the state of Washington, you could compare it to the state of Idaho, and you could get a sense of whether it's more risky to live in Idaho versus Washington for that disease. It's obviously very crude because lots of things are not being taken into account, right? You're not sort of like comparing people who are exactly like you and eat exactly the way you do and have the same lifestyle as you. Case fatality rate is a metric that tells us how likely you are to die of a disease if you actually do contract it, right? So it's sort of more looking at like the risk of dying if you do get sick. How dangerous is this disease? So hopefully that's clear. If we want to calculate these numbers, we're not going to be able to do it with something that's non-lethal, because when these 30,000 students were exposed to the cold and there were 600 people who had the cold, nobody died. So we really need to look at a different example. These are real numbers from the pandemic. In our Snohomish County, population 822,000, on Tuesday, June 2nd, 2020, there were 2,987 confirmed COVID cases, and by the next day, this number was 3,016, with a total of 150 deaths. So let's start again with morbidity. On Wednesday, what's morbidity? On Wednesday. The morbidity, Taylor, 2,987 is the morbidity on Tuesday. What's the morbidity on Wednesday? That's right. Morbidity is 3,016, mortality 150. Prevalence, how do we calculate prevalence? Prevalence is our total caseload divided by the population times 100, 0.36%. What about incidence? I'll give you a second to calculate incidence. If you want to put your answer into the chat, let me rephrase that. You must put your answer into the chat. If you have no idea how to calculate it, put a question mark. You have to put something. And if you want to show your work, that would be lovely. I'll give you some time to think about how to do this one. What is the incidence? How do you calculate incidence in this case? On Wednesday, we need new cases divided by population times 100. We just calculated prevalence, which is total cases over population times 100. What is incidence? And again, if you don't know how to do it, you can put a question mark. Well, look at that. Taylor has given us a beautiful example of how to start this calculation. So she said to find new cases, she took the current total cases on Wednesday, subtracted the number on Tuesday. That gave us the new cases, right? New cases, divided by the population times 100 and gives you this percentage, right? And she rounded it up to 0.004, 0.0035. Yep. Good job, everyone. This is exactly right. Now, the mortality rate, you take your deaths over population times 100, that gives you this number. Okay. Now, I'm going to calculate the case fatality rate just so that, you know, because it's something I think is interesting to know. But again, I just want to remind you, this is not on the exam. Case fatality rate, how you do this is you take the cases divided by the new case, you take the deaths divided by the total cases times 100 and gives you case fatality rate, sort of a measure of how likely you are to die if you do get sick. Okay, so deaths over total cases times 100. All right. Are these numbers accurate? What do you think? Are these numbers completely accurate? Taylor says no, Kate says no. And what is the reason that it is unlikely to be accurate? Taylor says not all cases reported. Yes, not all cases being reported. Are there people who have gotten coronavirus and it didn't show up in the official record? For sure. Are there people who died and it didn't show up in the official record? For sure. Okay. That is a big problem. But with that said, we still do the work, to hope to get a good enough idea of what's going on that we can still make good decisions. All right, Arizona State, a few months later, population 7.3 million. At that point in the pandemic, 188,737 total cases, 1,214 new cases that day, 4,199 deaths. So again, we want to have all these calculations. So let's start with morbidity. What's morbidity? On Tuesday, August 11th, what's morbidity? Yes, the big number. Morbidity on that day in August is 188,737. Yikes. What's incidence? Calculate incidence and put your answer into the chat. And then for this one, I'll wait. If you don't have any idea how to do it, put a question mark. But I'm not moving on until I see something in the chat from everybody. What is incidence on Tuesday, August 11th in Arizona? And if you're feeling unsure, you can put a question mark, or you can read the chat. You can copy other people. That is all good. Great. Okay. See, we just keep working it and we all work together and it gets better, right? Okay, wonderful. So morbidity, 188,737, mortality, of course, 4,199. Prevalence, I'm just showing you the math, would be to take total caseload divided by population times 100, right? That's going to be 2.6%. And incidence, as many of you calculated, you take the new cases over the total population times 100.016%. All right, now if we wanted to calculate mortality rate, we would take deaths over population times 100.057%. And if we want to calculate case fatality rate, we take deaths over cases times 100 for 2.2%. And just a reminder, what is this telling us? Case fatality rate, how likely are you to die if you catch this disease? 2.2% in this case. Mortality rate, what is your risk of dying from this disease just because you live in Arizona on Tuesday? 0.06%. And then what's the new case load over the per capita, and what's the total caseload per capita? Which of these statistics do you think would be the earliest indicator of change, worsening or improvement, if you were to watch them on a daily? Which one do you think is the most sensitive to change? To give us a sense of whether things are getting worse or better. I agree with you. You guys are on it. I would say for sure. And I can tell you from my personal experience that incidence was the thing I was watching for 2.5 years every day. What's going on with incidence? Okay. So if we compare Snohomish County, which was one of the early epicenters of the disease in the pandemic, in this country at least, if we compare Snohomish early in the pandemic to Arizona a little bit later, we can kind of just line up all these different metrics. We can say, well, there's our total population. And we can start to understand why population and the fractions are so helpful, because if we just look at total caseload, it's hard to know what does this mean? Because this could mean good or bad, just depends on the total population. So incidence is helpful in that case, prevalence is helpful in that case, and then things like mortality rate. So if we look at all these numbers, what would you say? Where is the outbreak worse? I don't think there's one right answer, honestly, actually, but I think there's a better answer. But just give us a vote. Okay, everyone has to vote. Snohomish or Arizona? Where would you rather be? Snohomish in June or Arizona in August? Where would you rather be? And I'll wait for everyone to weigh in. So everyone has to vote. Okay, great. So tell me why. What's the reason? It doesn't have to be a completely well thought out, but just what was the thing that tipped you over? The one thing you're looking at, the most compelling reason is blah. The statistic on yada yada is okay. Lower new cases, says Taylor. Prevalence is higher in Arizona, says Darsh. Oh, Eunice says she'd rather be in Arizona because the CFR is lower. Yeah. So like I said, there's not just one right answer here. Good job. So here's the thing, right? You can look at a couple different metrics. You can see different things. You can see that incidence and prevalence are considerably lower in Snohomish. This kind of makes sense, though. It's earlier in the pandemic. It just hasn't spread as much. It could, however, indicate that Arizona is doing a poor job of containing it, which was true. They did a poor job of containing it. We can look at mortality rate. What's your risk of dying, right? Out of the total population, fewer people are affected and fewer people are dead in Snohomish. The case fatality rate is worse. That looks pretty damning, and there could be lots of reasons. As it happens, this is quite common early in a disease for the CFR to be inflated because there's fewer data points and because the people who are showing up in the hospital are the more severely affected. All kinds of things happen later, right, in the disease outbreak where you get better tracking, you get better testing, you get better treatments, right? So it could be real. It could be that actually if you do get sick, you get better treatment, or it could be an artifact of the difference in the population and the difference in tracking at that time. Okay, now in terms of accuracy, when we look at these numbers, where is it likely more accurate? I mean, probably where you have more data points, but it depends on how good their tracking is. Are cases going up or down or stabilizing? Well, this is just a point in time. We don't really know, but if we were to watch incidents, we'd be able to tell. If we think about what's happening right now today, are things better or worse than they were at these different points in the pandemic? And if most people are saying better, well, everyone is saying better. Why? What's your reason? There's not just one right answer again, but what's the reason things are better? Incidence rates are lower, immunity has built, new cases per day is down, there's more herd immunity, lower deaths. And what is the reason for that? Why are there fewer deaths? There's actually, I would not say that the caseload is down. We went through a period just recently where like everyone under the sun, it seems like, came down with coronavirus. I didn't know anybody who died from it this time around, though, unlike the first time. What's the reason? Vaccination and treatment, right? Yeah. So we have a much better tools now at this stage, having fought this and sort of learned about this disease for several years. Okay. So let's just have a look at our time. I don't want to miss our break. Just hold on. Oh my goodness. There we go. 5:55. Okay. 17, 4. I will keep going just for a few more slides and then we will have a break in a minute or two. Here we go. So we also defined some terms earlier, endemic, right? Meaning that endemic is a term that's referring to a disease constantly present in a population, things like the common cold or the flu. Epidemic, as many of you mentioned earlier, is an unusually large case number. So it can either be an epidemic can be from an introduced disease where previous cases were 0. So anything over 0 is going to be an epidemic. Or it can be from an endemic disease that has a normal baseline and we're just seeing cases that are higher than that. So the flu goes through epidemic periods, even though it's also endemic. So you can be both things. You can be both endemic and epidemic, or you can be an introduced disease and epidemic. And pandemic, of course, is a worldwide epidemic. Now, which of these terms applies to COVID? I think you'd have to have been living under a rock not to recognize that it's a pandemic. What is the prediction in the future? Or maybe you might feel like it's happening right now. Yeah, the prediction is that it's going to become endemic, that we will have, in addition to our cold and flu season, we're going to have the coronavirus season, right? Lots of people get it, but it's pretty common. Everyone gets it. Okay, so I am going to skip over this question and move on to herd immunity. The definition that we talked about in the beginning was what? When most of a population is immune, it protects the unvaccinated or non-exposed remainder. What is the gold standard? What are we aiming for usually? Put your answer into the chat. We got 3 answers so far, waiting for a couple more. So there's not one right answer, but in your curriculum, sort of the ideal is around 90%. Now you can get herd immunity a couple of ways. You can be exposed to something and it can go through the whole population naturally. And this is how we used to try to get herd immunity for chickenpox. I don't think any of you are probably old enough for this to be the case, but when I was growing up, there was no chickenpox vaccine. So you would just try to get infected from other people. That was pretty standard. And vaccinations reduce that need for that, or that happening, because you can just be vaccinated and develop immunity without actually having to get chickenpox, which is much preferable because, as it turns out, and this is, I think, something that wasn't well known when I was a child, if you get chickenpox naturally, what happens is it goes latent, stays in your body, and can become shingles later in life. Okay. The flu is, of course, a respiratory virus that we're all very familiar with. Annually, a good percentage of the global population catches it, and then 500,000 people die from it on an average year, as compared to 1918, when about 20 to 50 million people died from it. So normally the flu would be characterized by which of these terms? That's right, endemic. How would we describe 1918? The 1918 flu. Yeah, just like coronavirus, it was considered a pandemic. Okay. I'm going to skip over this because it's just practicing some fun things. Let's talk about herd immunity for a second. Population of Snohomish, Michigan, 822,000. I am rounding. Flu vaccines are between 20 to 65% effective. The flu vaccine is administered annually. They re-up it. So you can get like the 2018 or the 2019 or the 2020 version. And its effectiveness varies from year to year. 2018 was not a very good year for the flu vaccine. It was only 25% effective. 2019 was better, it's 45%. How many flu vaccines needed to be administered to get herd immunity from flu in 2019? Think about it for a minute and we'll talk about it. Remember what the goal is we want 90%. 90% immunity. Excuse me. Okay. So typically people will give me an answer that is 90% of this number, because 90% of the population is what we want for immunity. The problem with the flu vaccines is that they're only 45% effective. Even if you administer them to every single one of these people, you're actually only going to get at most 45% immunity. So that is why it's difficult to control the flu with vaccination. However, is it therefore useless to get the flu shot? You are correct, it is not useless. And what the data shows is that getting the flu shot reduces total caseload about 50%, which is still quite useful because it helps to reduce the amount of pressure put onto the emergency room and to the medical staff and during each flu season, especially a bad flu season. Okay, so we're going to take our break now, and after the break we're going to do some more work. But let's see, I usually take a 10-minute break. I'm just going to have a look at our time. It is 6:03. Is 7 minutes long enough? Can we come back at 6:10? Perfect. All right, we'll come back at 6:10. I'm just going to suspend the recording in the meantime. All right, welcome back, everyone. I hope your break was relaxing. 7 minutes, woo, the fun. Let's move on. So at the beginning, we talked about nosocomial. The definition was a disease acquired in the hospital setting. It needs its own term because of the sort of unique predisposing circumstances in medical facilities. It's a little counterintuitive, I think, for people on the street to think that you are putting yourself at risk of disease by going to seek medical care. But that's, because you are, if you're in a hospital environment, you are likely to be exposed to people who are sick with things. You are also surrounded by people who have a higher level of compromised immune system, so they're more likely to be able to catch things. There is a chain of transmission that can occur between patients as a result of movement of staff between patients. And there's resident microbes that start to live inside of the hospital. And it's important to be aware of this so that we can know what the most risky behaviors are and how to sort of reduce risk for patients. When we look at the types of infections that people get, there's sort of a pie chart here. The most common type of nosocomial infection is a UTI. Is there anything that you know that happens inside of hospitals that would explain this? What is a procedure or an experience? What's the reason for this being one of the most common nosocomial infections? Oopsies. Put your answer into the chat. Eunice is right. catheterization. If we look at the types of procedures that lead to nosocomial infections most frequently, we see that urinary catheters and IV catheters and surgery and ventilators, all these things that basically can bypass your natural defenses. It makes sense that UTIs could be caused by urinary catheters because you're kind of making a little highway that microbes can crawl on so that they can go up into your urethra. It makes sense that if you cut people open or if you give them IVs, that you are penetrating the skin and introducing a pathway by which microbes can invade the body. With that said, it's interesting to consider that not everybody catches a disease if they're exposed, right? And not everybody gets a nosocomial infection. So there are certainly predisposing factors from the individual perspective, like as an individual person. Can you think of anything that would change your likelihood, your personal likelihood of getting a disease? Something about, okay, so genetics, age, physical condition, all these things are important. How good is your immune system? Yeah, absolutely. And of course, there's physiology. Disproportionately, women are more likely to have a UTI than men because their urethra is shorter. So it's shorter distance for the microbes to travel. Okay, so it's certainly the case that individual factors can account for some of the differences. That's not the only thing to think about, especially from an epidemiological standpoint. If we think about things like what types of things people die of, in the early part of the last century, the leading causes of death were all microbial pathogens, either causing respiratory or GI tract illnesses. And the demographic of death was in infancy or toddler age, small children. Currently, the leading causes of death are so-called old age diseases, things that you die of once your body is worn out. None of them are infectious disease, although if you looked at this during the pandemic, coronavirus was within the leading causes of death, but that's fairly rare now. This change is not bear out when you look globally, however. So if you look at the entire world's population, it's still the case that more likely than not, you're going to die at a young age of respiratory or GI tract infection. So young children still bear the brunt of disease globally. And so when we think about what those reasons are, right, it must be the case that there's more than just individual factors that account for that. There's environmental and geopolitical and social factors that account for those differences. If we think about what's changed in the United States in that 100 years, would it be true to say that antibiotics, antiviral drugs, vaccines, and medical advances have made the largest contribution to reducing deaths from microbial infection? Is that true or is that false? I think that if you were to ask almost anybody, they would say, sure, this is true. It's medical interventions that have changed the course of mortality from infectious disease. It turns out not to be the case, however. Certainly, it's an important contribution. But if we look at the total death rate in the United States, this is actually 1890, the total death rate in the United States has gone down over the last 100 and some years. But if we look at when the things like vaccine use, antibiotic use, when those occurred, penicillin, the first antibiotic, wasn't widely deployed until the 1940s. Widespread vaccination campaigns didn't come about until the 1950s, starting with polio. So there's a lot of reduction in mortality that's happening here. That's the result not of medical intervention, but of epidemiological intervention, establishing public health departments where people were paying attention to patterns, treating the water, so basic infrastructure change so that drinking water is safe, and a variety of other things that are not shown here, but getting the trash collected, treating wastewater, getting the word out that you should wash your hands, stuff like that. Well before these medical interventions, which obviously were important. Look at that, they're still causing a reduction in death. But if we're looking at like the biggest impacts are being made, at least in the US, by these other non-medical interventions. So that's really interesting. And that's not to say that vaccines aren't important and medicine isn't important, because when we vaccinate people against measles, measles cases go down. When we vaccinate people against pertussis, pertussis cases go down. But if we're looking at what are all the factors that are involved in making a society well, medical intervention is really only part of the picture. And if you watch the video lectures for this section, you'll know that all of these are really important factors that affect your likelihood of becoming ill, depending on where you live. How is the water? Is it safe to drink? What's sanitation like? Is there waste kind of spilling out in the street? Is there a lot of air pollution? Is there a lot of water pollution? Are there chemicals that will make you ill? Is the air safe to breathe? Are you getting enough food? Do people get a good education? Do they have enough resources? Is the government corrupt or not corrupt? And is the infrastructure intact? All of these things are really important. And we can really illustrate this point with one sort of, I think, disease story. Because if you ask someone right now, like, where are you at risk of getting malaria? Tell me like one part of the world that comes to mind when you hear this word. Write that into the chat. Where are you going to go if you're like, oh, I have to worry about malaria? Yeah. And that's true right now, Africa and Asia. But 100 and some years ago, this is 1882, in the US, malaria was really common. This is 50 years later, 1932. Now this is where malaria is relegated to. And it's really not an issue anymore. So what's the difference? What happened in those 50 years? Well, one of the things that happened were massive campaigns to reduce standing water, and also massive campaigns to spray chemicals that would kill mosquitoes. It was very effective, okay? Was it perfect? Not by a long shot, especially when we look now at some of the consequences of these activities. These drainage activities, which reduced swamps and standing water, also impacted wildlife and the sort of ecological health of the planet and the parts of the world where this was happening. And pesticide use has driven the rise of pest resistance in microbes. So climate change also is causing new habitats to open up for mosquitoes. And because of the success of this in the US, it's been deployed in other places, which has led to even more pesticide resistant and even more damage to the environment. So it's complicated, right? If we look at disease as a whole, again, as a global question, we certainly in no way can say that disease has been eradicated or solved. And part of that is, of course, because new diseases arise all the time, such as coronavirus. Okay. So I'm going to skip over these slides because it's just a fun way to look at these numbers that we've been doing again and just talk about emerging diseases and the factors behind them. Let's first look at some of the successes, right? Diseases entirely or nearly eradicated in the last 100 years. Can you think of any? Put them into the chat. If there's any disease that you were like, yeah, 100 years ago, this was really a problem globally, and now it's not. Yeah. Great examples, guys. Yeah. Here's some of mine. Some of you mentioned these, some of you mentioned other ones, which are good examples. Smallpox, though, sort of the poster child for this, for sure. A massive vaccination campaign, a global vaccination campaign in the 70s eliminated it from the planet. A disease that had been in the human population for like basically as long as we have records, and probably before we have written records of any kind. The Black Plague, the bubonic plague, the Black Death, they're all the same. A bacterial infection, which is now well controlled with antibiotics, and we now understand how it spread, and so we can reduce that. Polio, there were some surges of polio in the US that were well controlled subsequently by vaccination. Tuberculosis, another bacterial infection well controlled with antibiotics, at least initially. And many, many STDs, most of which were bacterial, which were well controlled with antibiotics. Now, with that said, in the last 50 years, we've had lots of new diseases come on the scene, right? So I would argue way more new ones than we have been successful at beating back. So Ebola, HIV, different strains of E. coli than we'd seen before, prion disease, mad cow, all kinds of coronaviruses, starting with SARS and MERS, and now coronavirus, COVID-19, a variety of mosquito-borne infections, and cholera, which was initially a success in terms of understanding like how you save people. It's very easy to save people who have cholera. You just give them electrolytes, basically. But if you can't do that, well, then they will die very quickly and horribly. Now, and it's not only the case that we have like these newly emerging diseases that we've never seen before, but diseases that previously were well controlled also have come back. So multi-drug resistance tuberculosis is now a thing, multi-drug resistant gonorrhea, multi-drug resistant staph. We're running out of antibiotics to treat these bacterial infections for all variety of reasons. And when I was growing up, the finger was always pointed at individual patients who were taking the antibiotics wrong or asking for too many antibiotics. But it is becoming increasingly clear that a major contributor to this is also changes in how our food stream is produced. Animal production is frequently supplemented with low doses of antibiotics, which is the perfect scenario in which drug resistance can arise. Okay, so what are the factors that contribute to emerging and re-emerging diseases? Put that into the chat. Anything that would lead to a disease emerging on the scene or a previously controlled disease re-emerging. Okay, we have genetic recombination, says Taylor. Yes, so pathogens changing their genes, coming up with new toxins, new virulence factors, new ways to get into the human body. Climate change giving new habitats for pathogens so they're more widely spread. Modern transportation, says Julius, moving stuff around the world, like if your disease is endemic to, let's say, Venezuela, but not to the States, but then you get on an airplane and bring it to the States, there you are. Now you have brought it to a new place. Ecological disasters causing massive migrations of people to new areas, and those refugees often being in unsanitary conditions because their resources are, it's not ideal. Not using antibiotics correctly, animal control measures, overuse of pesticides, and certainly gain of function research on viruses, using what we know for evil instead of good. There, you can use the technology that we have developed to use microbes as bioweapons. That's a possibility. So all of these things contribute to the emergence of disease. All right, we're going to focus on antigenic change, and we're going to focus mostly on antigenic change in viruses, although this can happen in lots of contexts, including bacteria. We've already defined antigens a little bit. We're going to revisit this definition. Remember, we talked about it during pathogenesis, and we talked about antigens as being something that could invoke an immune response. from the perspective of the microbe, antigens are things on its surface that our bodies can recognize and build an antibody against. So it's anything that can be recognized by an antibody. Protein or complex carbohydrate is typically the type of chemical that's recognized by an antibody in the mammalian immune system. What are some things on bacteria that are made of protein or made of sugar that could potentially be antigenic, that once introduced into the human body, our bodies could notice, respond to? Yeah, the cell wall. Exactly right, Julius. For sure. The cell wall. It's made of protein and carbs. That's an antigenic feature. What else? Think about all microbes, not just bacteria, although certainly bacteria. Are there bacterial features that are made? Spikes on viruses? Yes, those are antigenic. For sure. Protein spikes on viruses. What else? Is there anything protein based on bacteria besides the cell wall? Is there anything carbohydrate based on bacteria besides the cell wall? Peptidoglycan, yep. DNA and RNA, interestingly, are not super antigenic. They're kind of boring molecules. Enzymes. Yes, enzymes are proteins, and if released, like exoenzymes, for example, made by bacteria, for sure. Yep. Although sometimes they're so similar to human enzymes that they don't cause an immune response, but they could if they're new, different. What else? Is there any structure on bacteria that's made of protein? Or any structure on the outside of a bacterium that's made of carbs? Yes, the protein capsid of a virus for sure could be antigenic. Yes, Darsh, that's a good one. Anything else? All right, good examples, everybody. So many of you mentioned bacterial cell walls. Also on bacteria, the proteins in the membrane, the capsule made of sugar, that could be antigenic. That's actually used in some vaccines. Viruses, as many you've mentioned, have spikes, capsid proteins. These are also antigenic. Protozoa are animal-like. They don't have a cell wall, but they do have membrane proteins. When we think about viruses, I think probably by now, not surprising that they have really short generation times and they can change pretty quickly. There's these two terms we're going to use to describe the types of changes that can happen. Both of them are genetic changes. Antigenic drift and antigenic shift. Just looking at these words, which one do you think drives evolution more quickly? Yes, I'd agree. Antigenic shift. Let's talk about what this means. So antigenic drift. This is when those antigens, and again, what are those antigens? They're going to be the membrane proteins. They're going to be the carbohydrates in the capsule. They're going to be the proteins on a capsid, or they're going to be the spike proteins on a virus, right? So if we're talking about viruses specifically, we're looking at those proteins that are on the surface of the virus. Antigenic drift is when the instructions for those proteins of the virus change by one nucleotide because of a mistake made during replication. That one nucleotide change might change an amino acid in the protein, and that might lead to a change in the structure of the protein that can be detected by antibodies. So antigenic drift is slow, relatively speaking, and small, and it happens during replication. Antigenic shift. Antigenic shift happens when an entire gene is added or removed or changed. And in viruses, this can happen when multiple viruses with different genetic instructions co-infect the same host cell and swap bits of their genome, basically. So we're going to look at an example of this. Before we look at that example, let's talk about the flu. And we're going to come back to the flu for this example. But why do we need a new flu shot every year? Why don't you have, like, there's lots of things you can get a vaccine for where it's sort of maybe one and a booster and done. What is the reason we have to have a new flu shot every single year? Yes, that's right, Julius. Different strains are circulating. Eunice says this is due to mutation. These are both very true. Now, the flu is a bit different from a lot of other viruses in that it has 3 factors driving its rapid evolution. The first is that it is an RNA-based virus, and RNA-based viruses use RNA polymerases. Often they use RNA-dependent RNA polymerase, which is a viral polymerase. And these are error-prone, which means every time they copy RNA to RNA, they make a lot of mistakes. This drives antigenic drift. Also, unusually, flu viruses typically have multiple hosts that they can get into. You might remember from the virus section that I told you that most viruses are very specific in terms of their host. But flu usually has multiple hosts it can get into. And finally, it has a segmented genome. It doesn't have one piece of RNA or DNA, which is more common. It has multiple little mini chromosomes, and these can be swapped if multiple flu viruses co-infect the same host. So this is just showing you that flu viruses often have multiple hosts. For example, there's quite a few human flus that can get into pigs. And there's quite a few bird flus that can also get into pigs. That makes the pig the perfect mixing ground for avian and human flu viruses. So we can imagine, and we don't have to imagine, has happened multiple times, but you can imagine that a flu virus that is got its surface spikes can't get into people, okay? And a human virus whose surface spikes can get into people can both get into a pig cell. And then when they're replicating in that same pig cell, they can mix and match their genomic material and create a Frankenflu, a new flu that's never been seen before, and is not different by one nucleotide due to replication error. It's different by a whole bunch of different genes. It can now get into people, but it has brand new other things about it that come from the bird. Okay. So one of the things that I like to do when I teach on Zoom is use the Kahoot quizzes, but in live time. And there's not very many of us, but I think it'll be still fun to do. Are we game for this? Would you like to do that? Julius is like, for donuts. All right. Well, give me a second. I'm just going to how do I stop? Dang it, how do I stop the share? There we go. I'm going to stop the share so I can go into my Kahoot account and get loaded. Well, I'm not going to drink or anything that's silly, but what you want to do is go to kahoot.it. Go to kahoot.it. If you already have Kahoot loaded up on your phone, you can also, or on digital device, you can go and just log in or just open that app up, and that'll also be good enough. I'm logging in right now. I'm going to pull up the pathogenesis Kahoot, and we're going to
Did you say that you were going to share your screen, what you see on the Kahoot, or?
I will in a second.
Okay.
Yeah, I'm just trying to get into the Kahoot right now. It's giving me a bit, but you can just bring the Kahoot up, and I will share my screen and give you the code in a second. Go into my library and start this pathogenesis and epidemiology Kahoot. I'm going to share my screen now. That will allow us to share. There we go. Thanks. Go into my. Please let me in. It's giving me all kinds of classic. That's what I want. All right. And I also want to share the music because it's so awesome. So on your screen, you can see the pin 1761911, and you're going to use that to log in to the Kahoot. So you're going to go to kahoot.it or open the Kahoot app, and you're going to put in this game pin, and we're going to play this game together. All right, so we got 4 out of 5 of our people. If you're not planning to play the Kahoot, and you can just let me know that in the chat, and I won't wait for you. Okay, looks like we got everybody. Let's do this. Here we go. Shigella has an ID 50 of 100. The ID 50 of Salmonella is 1,000,000. What does this mean? Right. Excellent. Yes, Shigella is more infectious. Shigella is more infectious. The ones that refer to it's more likely to kill you are not relevant because the infectious dose doesn't tell you about lethality. And it is more infectious because the ID50 is lower, meaning it doesn't take as much to infect you. True or false, the most important historical contributors to disease reduction have been antibiotics and vaccines. Historically, is this true or false? Think about the last few centuries. The game pin is 1761911. It's right at the bottom here. The most important historical contributors to disease reduction have not been antibiotics and vaccines. They have been infrastructure changes like developing public health departments and treating wastewater and providing clean, safe drinking water. And that's still the case worldwide. True or false, antibiotics can temporarily worsen symptoms and increase your risk of a secondary infection. That is true. All right, true or false, an asymptomatic individual, one who is showing no symptoms, can be a reservoir of disease. Also true, good job, you guys. True or false, you can predict the LD50 of a microbe if you know its ID50. That is false, good job. They are independent metrics. Shiga toxin has an LD50 of 250 nanograms per kilogram. If you gave 200 nanograms per kilogram to 50 people, what would happen? less than 25 people would die because we are under the LD50, right? If it was exactly 250, then we would predict 25 people would die. If it was more than 250, we would predict more than 25 people would die. All right, exotoxins include all of the following except which one is not an exotoxin? It is lipid A. Remember, that's the endotoxin. Koch's postulates were most important because for the first time, scientists could prove causation. They could prove causation. Lots of people were doing correlative studies at that time. Proving causation was the innovation that his postulates represent. Koch's postulates might not allow to establish a link between a pathogen and a disease under which circumstances? Yes, it is all of these. Good job, you guys. All right, this is a multi-select. There might be more than one right answer. Normal microbiota are found where? Skin and mucous membranes, correct. If it's in your bloodstream or internal organs, you are not in a healthy state. A hiker's skin knee fails to heal. Instead, they end up with sepsis. This is an example of what? It is an example of a focal infection. Started local, normally would have cleared, but somehow ended up systemic. True or false? Normal microbes can be opportunistic pathogens. It is true, guys, and you nailed it. Another multi-select. Which of the following would be symptoms of the flu? Select all that apply. All right, all of these except not fever, because fever is considered not a symptom, but a what? Write into the chat what it is. Not a symptom, but a sign. That's right, because it's objectively measurable. Okay. Sam was exposed to norovirus February 27th, had a stomachache the 29th, vomiting and diarrhea the 30th to the 31st. Which of these statements is accurate? There we go. It was like on a delay. So the prodromal period is February 29th, early mild symptoms. Incubation periods are all reported incorrectly. What is the correct incubation period? Put that into the chat. transmission of the first sign or symptom. And so what are the dates of that incubation period? What are those dates? What would one of these have to, it would have to be not, the 29th is that prodromal period. What's the incubation period? It is the 27th, 28th. And even if you had written 27th through 29th, I would accept that. It's exposure to the first symptoms. So exposure to 27th, first symptoms 29th. Good. All right. If 10 people in a class of 100 have a cold, the prevalence is what? %. Good job. We took our total cases divided by our population times 100 gives us 10%. If 10 people in a class of 100 have a cold, what's the incidence? It is unknown. Need more information. Why do we need more information? What don't we know? Put that into the chat. What do we need to know in order to do this math? We need to know new cases, right, Julius? That's right. We need to know how many of these 10 people newly acquired the cold in that time period rather than just have it. All right. Morbidity. What does it tell you? It is how many people are affected by a disease. It's just the total caseload. Good work. Multi-select. More than one answer might be correct. New diseases emerge all the time because all of these, right? Microbial evolution, climate change, human migration, and use of antibiotics and pesticides, all of these. Fomite transmission is transmission how? Touching a contaminated surface or object. So there's an inanimate intermediary in fomite transmission. Used Kleenex, stapler that someone sneezed on, door handle someone touched. So it's an inanimate, it could be like a floor, it could be a lot of things. Touching a contaminated surface or object is how we contract the disease. And last one, the parenteral route. Break in skin or mucous membrane, such as injection from a needle. Yes. The fun of Cahoots. You get to potentially be on the podium, but I know that y'all were running neck and neck. So congratulations to everybody for doing so well in our Kahoot today. All right. Let's go back to the slides just for a hot minute. I know that we are coming up on the end of lecture time. I can find my slides again. There we go. There's two things I wanted to say about the lab report, but I first want to make sure that you know what's happening next. So there's no lab tonight because of the fact that we are not on campus, and then campus is closed. So we're going to do finish lab 8 on Thursday, and we will do the ELISA as well. And I think we'll have time. Lecture will be applied immunology just like it was supposed to be. Typically doesn't take a whole 2 hours, so we should be able to fit the lab stuff we need to do in. Sunday, the epidemiology quiz is due, and lab 8. You'll have data on Thursday, so it still gives you time to do that. And the post lab 8 quiz, which you can do sooner really, because it's based on largely based on the things that you've done digitally. I want to remind you that the lab report deadline is coming up, and the first deadline is March 5th if you want to submit it early for extra credit. I do want to just briefly mention a couple things about that lab report, just to remind you the guidelines for what you need to do to submit it. There needs to be a results table that has the dates of the tests you ran, the tests themselves, and the observations. No interpretations. Do not tell us what the test results mean, just tell us what they look like. Kate says is March 1st, the early deadline, March 5th. March 5th is the early deadline, March 5th. That's Sunday. In the discussion, you need to make a flow chart, and then you need to, your first paragraph should tell us what metabolic tests led to your group, state what your group is, and then describe what you learned from the part 3 subsequent metabolic tests. Don't worry about sequencing. We don't have that part right now. And then you have a research section where you talk about what other people know about your species, and make sure you have at least 3 references. It needs to be, the whole report should be single spaced, and this research section should be 3/4 of a page. The references need to be peer reviewed and legitimate. I wanted to take a second to talk about the best fit analysis, because some of you may reach a point where you have completed the tests that you could do that make sense to do, and you still have a few species left. If that's the case, you can do something called best fit. Best fit analysis is when you've done every kind of test where your remaining species, there's at least 1 positive and 1 negative for that test. And now all the tests that you have left would be unhelpful. And so in that case, you can build yourself a little best fit table that looks like this. It's going to be much bigger, but mine has 4 just for an example. You're just going to put every test that you received, including ones that were inconclusive from the lab tech. And then you're going to list the actual result that you got for your unknown, and then the Bergey's table results for the other species. And then just go item by item. Some of the tests will not be helpful because everybody is the same, but in some cases you will notice differences. So for example, when we look at motility, you are positive. These 2 species were positive, but this one was inconclusive. So that's just one kind of like, doesn't quite match my data. The nitrate test, you were inconclusive, but this one actually was positive. So that doesn't really match my data either. Lysine doesn't really tell us anything. The xylose test, you were positive. This guy didn't have any data, so that doesn't quite match my data. And so out of these, this one would be the one that is the so-called best fit. So March 5th is the early extra credit deadline. If you want extra credit, you can submit early. And the final deadline is the following week. Okay, at this point, if you want to leave, you can. You just would sign out. But I will stick around for a few minutes until everyone is gone, because if you have specific questions about any of this material or any upcoming assignments or the unknown ID lab report, I'm happy to answer them. But for right now, I'm going to stop the recording so we can post it.